There is a world beneath our notice, a world that moves in shadows and sunlight, a world so small and yet so vast that it contains multitudes. And tonight, we step into that world, not as giants, but as quiet observers, watching, listening, noticing the intricate machinery of life that unfolds on scales almost too small to see. Imagine a meadow somewhere distant and quiet, where the grass grows tall and soft, where wildflowers bloom in scattered patches of color, purple and yellow and white, and where the air hangs thick with the scent of earth and pollen. This meadow exists beyond the reach of cities, beyond the hum of traffic and the glow of screens. And in this meadow, time moves differently, measured not in hours or minutes, but in the opening of petals, the arc of the sun, the slow unfurling of wings. We are here to witness something extraordinary, something that has been happening for hundreds of millions of years, long before humans walked upright, long before we built our first shelters or spoke our first words. We are here to observe the insects, those six-legged architects of the natural world, those tiny engineers and gardeners and hunters whose lives unfold in a complexity that rivals our own. The word itself, entomology, comes from the ancient Greek entomon, meaning that which is cut in sections, a reference to the segmented bodies that define these creatures. 3 parts joined together, head and thorax and abdomen, each section a marvel of biological engineering. And the study of these beings, the science of entomology, is the study of persistence, of adaptation, of survival against impossible odds. In this meadow, if we lower ourselves down to where the grass stems tower like trees, we begin to see them. A beetle moves slowly across a leaf, its shell gleaming like polished armor in shades of emerald and copper. Each step is deliberate, methodical, its antennae sweeping the air, reading chemical messages we cannot perceive, testing the world through senses we can barely imagine. Nearby, a butterfly rests on a blade of grass, its wings folded upward like hands in prayer. The wings themselves are miracles, thousands upon thousands of tiny scales, each one a single color, arranged in patterns that create the illusion of smooth gradients, of perfect symmetry. When the wings open, slowly, carefully, they reveal oranges and blacks and whites, warnings to predators, or invitations to mates, or simply the result of millions of years of trial and error, of wings that worked and wings that didn't. The air above the meadow shimmer with motion. Dragonflies patrol invisible territories, their 4 wings beating independently, allowing them to hover, to dart backward, to change direction in the span of a heartbeat. They are hunters, these dragonflies, ancient hunters whose ancestors flew above the swamps of the Carboniferous period, when the air was thick with oxygen and insects grew to impossible sizes, when dragonflies had wingspans as wide as a hawk's. But here, now, in this meadow, the dragonflies are smaller, more delicate, and yet no less magnificent. Their compound eyes, massive and multifaceted, contain thousands of individual lenses, each one capturing a fragment of the world, piecing together a mosaic of movement and light that lets them track the smallest flying insect, calculate its trajectory, and intercept it in midair with a success rate that would shame any human-made machine. And beneath all of this, beneath the flowers and the grass and the visible creatures of the day, there exists another world entirely. The soil itself is alive, teeming with insects we will never see. Beetle larvae curl in chambers of earth, white and soft and blind, slowly consuming decaying matter, transforming death into the nutrients that will feed new life. Ants tunnel through the darkness, creating cities underground, chambers for their young, chambers for their food, highways and byways connecting it all. A civilization that operates on chemical signals and instinct, on roles assigned before birth, on cooperation so complete that the colony itself becomes a kind of superorganism, each ant a cell in a larger body. This is the world we are entering, slowly, carefully, with reverence for its complexity. This is the realm of the small, the overlooked, the essential, and we are here not to conquer or catalog, but simply to witness, to understand, to appreciate the strangeness and the beauty of these lives that unfold in parallel to our own. The evening light begins to soften, turning golden, and the meadow transforms once more. Different creatures emerge, moths beginning their first tentative flights, drawn toward the fading light, their wings dusty and soft, where the butterflies were crisp and bright. And somewhere in the grass, a cricket begins to sing, and then another, and another, until the whole meadow fills with that ancient sound, that rhythm that has marked the coming of night for countless millennia. We have only just arrived, and already there is so much to see. We might begin, as all things begin, with curiosity, with the simple human impulse to look closely at the world, to wonder about the creatures that share our space, that crawl across our paths, that fill the air with their humming. For most of human history, while insects were simply there, part of the background, noticed only when they helped or harmed, when they pollinated crops or devoured them, when they produced honey or spread disease. But somewhere in the distant past, someone paused. Someone watched an ant carrying a burden twice its size, or noticed the perfect geometry of a honeycomb, or wondered why a firefly glowed. And in that pause, in that moment of genuine curiosity, the seeds of entomology were planted. Though the formal science would not emerge for thousands of years, the ancient Egyptians knew insects intimately. The scarab beetle, sacred and symbolic, appeared in their art and their religion. They observed how these beetles rolled balls of dung across the sand, how they buried these spheres in the earth, and how, impossibly, new beetles emerged from the ground weeks later. To the Egyptians, this was resurrection, rebirth. The beetle became a symbol of the sun god Ra, rolling the sun across the sky just as the beetle rolled its precious cargo across the desert. They could not have known, these ancient observers, that the female beetle was laying eggs in that ball of dung, that the larvae would feed and grow in that buried nursery. That metamorphosis would transform the grub into the gleaming adult. They saw only the mystery, the miracle, and they wove it into their understanding of the cosmos. The ancient Greeks turned their philosophers' minds to insects as well. Aristotle, in the 4th century before the Common Era, examined insects with the same careful attention he gave to all living things. He dissected them, studied their structures, tried to understand their purpose in the natural order. In his History of Animals, he described bees and their societies, noting the presence of a leader bee, what he called the king. Though we would later discover the ruler of the hive was always a queen, Aristotle believed insects were simpler creatures, born not from eggs or live birth, but from spontaneous generation, from rotting meat or morning dew, from the earth itself. It was an understandable error. When maggots appeared on dead flesh, seemingly from nowhere, what else could one conclude? The truth, that flies laid invisible eggs, that life always came from life, would take nearly 2,000 years to prove conclusively. Still, Aristotle's work mattered. He established the idea that insects were worthy of study, that their small lives contained lessons about the natural world. He noticed the metamorphosis of butterflies, the organization of ant colonies, the construction of spider webs. Even if he misunderstood their origins, he recognized their complexity. The Romans inherited this curiosity. Pliny the Elder, in his massive natural history, compiled everything known about insects in the 1st century of the Common Era. He described silkworms, those caterpillars whose cocoons produced the most prized fabric in the ancient world. He wrote of bees and their honey, of locusts and their devastating swarms, of the purple dye extracted from certain sea snails. His work was less scientific than Aristotle's, more encyclopedic, a gathering of facts and folklore, truth and superstition mixed together, but it preserved knowledge, kept it alive through centuries of change. And then, for a long time, the careful study of insects seemed to pause. The medieval period brought other concerns, other focuses. Insects appeared in bestiaries, those illuminated books of beasts, but they were usually symbolic, moral lessons dressed in 6 legs and wings. The ant represented industry. The spider represented the devil's snares. The butterfly represented the soul. They were metaphors more than subjects of genuine inquiry. But beneath the surface of formal learning, knowledge continued. Farmers understood the rhythms of insect life, knew when the locusts might come, when to expect the beetles that ate their grain. Beekeepers developed sophisticated methods for managing hives, for harvesting honey without destroying the colony, for understanding the cycles of swarming and the seasons of plenty. This was practical entomology, born of necessity, passed down through generations, not in books, but in practice, in observation. In the slow accumulation of working knowledge, the Renaissance changed everything, slowly at first, then faster. The printing press meant knowledge could spread, could be shared and debated and built upon. The development of better lenses, of early microscopes, meant the small world could be magnified, revealed, seen in detail that would have seemed magical to earlier generations. In 1665, a man named Robert Hooke published a book called Micrographia. It contained illustrations of insects and other small creatures as seen through his compound microscope. The drawings were extraordinary. A flea appeared as large as a cat, every segment of its body visible, every hair, every joint, the compound eye of a fly, shown in exquisite detail, revealed the truth of how insects saw the world, not through single lenses like our own, but through hundreds of tiny facets, each one an eye unto itself. Hooke's work did something revolutionary. It made the invisible visible. It proved that insects were not simple, not primitive, but complex beyond imagining, that a creature smaller than a fingernail could contain structures as intricate as any found in larger animals. The book became a sensation. People who had never thought twice about insects suddenly wanted to look closer, to see for themselves these hidden wonders. At the same time, across Europe, naturalists were beginning to challenge old assumptions. Francesco Redi, an Italian physician, performed experiments in the 1670s that finally disproved spontaneous generation, at least for larger organisms. He placed meat in jars, some covered with gauze, some left open. The open jars produced maggots. The covered jars did not. The conclusion was inescapable. The maggots came from flies, from eggs laid by flies, not from the meat itself. It seems obvious now, but at the time it was radical. It meant that every insect, every living thing came from parents, from eggs or live birth, from an unbroken chain of life stretching back through time. It meant the world was even more orderly, more knowable than previously thought. Jan Swammerdam, a Dutch naturalist, took this further. He spent his short life. He died at 43, studying insect anatomy and development. He was particularly fascinated by metamorphosis, that seemingly magical transformation that turned caterpillars into butterflies, grubs into beetles. He dissected caterpillars at different stages, carefully, precisely, and discovered something remarkable. Inside the caterpillar, folded and compressed, the future butterfly already existed. The wings were there, miniature and crumpled, the legs, the antennae, all waiting. All metamorphosis did was allow these structures to unfold, to grow, to become functional. This challenged the idea that metamorphosis was a kind of rebirth. Instead, it was development, predetermined and programmed, unfolding according to rules written in the very structure of the creature. Swammerdam's work was so detailed, so careful, that much of it remained unsurpassed for more than a century. Maria Sibylla Merian, born in 1647, brought art and science together in ways that had never been done before. She was a botanical illustrator, but her real passion was caterpillars. She raised them, fed them, watched them transform, and she painted every stage of their lives with scientific accuracy and artistic beauty. Her paintings showed the host plants, the caterpillar, the chrysalis, the adult butterfly or moth, all together, demonstrating the connections between insects and the plants they depended on. In 1705, at the age of 52, she published Metamorphosis Insectorum Surinamensium, documenting the insects of Suriname, where she had traveled specifically to study tropical species. The book was revolutionary. It showed insects not as isolated curiosities, but as parts of ecosystems connected to plants, to other insects, to the environment. It was ecology before the word existed, but Merian faced obstacles her male contemporaries did not. Her work was sometimes dismissed, attributed to the men who sold her paintings, or ignored entirely by the formal scientific establishment. Still, she persisted, and her illustrations became the gold standard used by naturalists for generations, proving that close observation, patience, and artistic skill could reveal truths that dissection and experimentation alone could not. By the early 1700s, entomology was becoming a discipline. Natural history societies were forming. Collections were being assembled. Wealthy patrons sponsored expeditions to bring back exotic specimens from Asia, Africa, the Americas. Insects were being classified, named, organized into groups based on their characteristics. The sheer diversity was overwhelming. Every expedition brought back hundreds of new species, thousands. There seemed to be no end to the variety of forms that insects could take. Carl Linnaeus, the Swedish botanist who created our modern system of taxonomy, included insects in his great work, Systema Naturae, first published in 1735. He grouped insects by their wing structures, by the number of wings, by whether those wings were hard or soft, covered or exposed. His system was imperfect. It grouped unrelated insects together based on superficial similarities, but it provided a framework, a common language that naturalists across Europe could use to communicate about what they were finding. The 1800s would bring even better microscopes, the theory of evolution, a deeper understanding of insect life cycles and behaviors. But here, in these early centuries, the foundation was laid. The question had shifted from whether insects were worth studying to how best to study them, from what they symbolized to what they actually were. And in meadows and forests, in gardens and fields, the insects themselves continued their ancient patterns, oblivious to human observation, living out their brief lives with an efficiency and purpose that would inspire wonder in anyone who paused long enough to notice. The 19th century arrived with new tools, new ways of seeing, and an explosion of curiosity about the natural world that would transform entomology from a gentleman's hobby into a rigorous science. The Industrial Revolution was changing landscapes, factories rising, cities expanding, but it also brought precision instruments, better glass, microscopes that could reveal structures no human eye had ever witnessed. In laboratories across Europe, entomologists were discovering that insects possessed abilities that seemed to defy the laws of physics. The flea, that tiny pest, could jump more than 100 times its own body length. If a human could do the same, we would leap over skyscrapers. The mathematics of this were staggering. The flea's legs contained a protein called resilin, more elastic than rubber, storing energy like a compressed spring and releasing it in an explosive burst. This was biomechanics before anyone had coined the term. Charles Darwin, watching the natural world with his uniquely patient eye, saw in insects proof of his revolutionary theory. In 1862, he published a paper on orchids and their pollinators, demonstrating how flowers and insects had evolved together. Each orchid species had a shape, a scent, a color pattern designed to attract specific pollinators. And the insects, in turn, had developed tongues, body shapes, behaviors that allowed them to access the orchid's nectar. It was coevolution, a dance of mutual adaptation stretching back millions of years. Darwin predicted, based on the structure of a Madagascan orchid with a nectar tube nearly a foot long, that there must exist a moth with a tongue of equal length. His contemporaries scoffed. Such a creature seemed impossible, absurd. But 41 years after Darwin's death, in 1903, that moth was discovered. Its scientific name, Xanthopan morgani predicta, the last word meaning predicted, tribute to Darwin's insight. The moth's proboscis, coiled beneath its head when not in use, could unfurl to nearly 12 inches, reaching deep into the orchid's throat to drink nectar no other creature could access. This was the power of close observation combined with theoretical thinking. Darwin had never seen the moth, but he understood the logic of evolution so deeply that he could predict its existence. And entomology provided endless such examples. Every adaptation, every bizarre structure or behavior made sense when viewed through the lens of survival, of reproduction, of the slow grinding wheel of natural selection. Jean-Henri Fabre, a French naturalist born in 1823, took a different approach. He was not interested in classification or theory. He wanted to watch insects live, to observe their behaviors in natural conditions for hours, for days, for entire seasons. He built an outdoor laboratory, a plot of land he called his laboratory of the living, and there he studied wasps and bees and beetles with a novelist's eye for detail. Fabre discovered that the hunting wasp possessed a surgical precision. The female wasp, preparing a nest for her young, would hunt specific prey, a cricket or a caterpillar or a grub, depending on her species. When she found her target, she would sting it, not to kill, but to paralyze. The sting was delivered to exact points in the prey's nervous system, shutting down movement while keeping the victim alive. Fresh meat for her larvae. She would then drag this paralyzed creature to her burrow, lay a single egg upon it, and seal the chamber. When the wasp larva hatched, it would feed on the living, paralyzed prey, eating in a specific order, consuming the non-essential organs first, saving the vital ones for last, keeping its food alive and fresh for as long as possible. It was grotesque, efficient, and utterly fascinating. Fabre described it all in prose that read like poetry, making insects into characters, giving their lives narrative weight. His contemporary critics, the laboratory scientists, dismissed his work as too anecdotal, too literary, but Fabre understood something they missed. That behavior was as important as anatomy, that watching how an insect lived revealed truths that dissection never could. His books became bestsellers, translated into dozens of languages, inspiring generations of naturalists to look more carefully at the small dramas unfolding in their own gardens. The late 1800s brought another revolution, the understanding that insects could be vectors of disease. For centuries, people had known that swamps bred illness, that crowded cities suffered plagues, but the connection to insects was unclear. Ronald Ross, a British doctor working in India, proved in 1897 that mosquitoes transmitted malaria. He dissected mosquito after mosquito, examining their stomach walls until he found the malaria parasite developing there. The implications were staggering. Suddenly, insects were not just curiosities or agricultural pests. They were matters of life and death, of public health, of empire and war. The Panama Canal, that massive engineering project, failed in its first attempt largely because yellow fever and malaria killed thousands of workers. It succeeded years later only after aggressive mosquito control measures, draining standing water, screening buildings, eliminating breeding sites. Entomology became strategic, essential, funded by governments and military organizations. But even as insects were being studied for practical reasons, others were discovering their extraordinary sensory worlds. Augustus Forel, a Swiss myrmecologist, devoted his life to understanding ants. He learned that ants communicate primarily through chemical signals, pheromones, leaving trails that others can follow, marking territory, identifying nestmates, coordinating group activities, all through sense we cannot perceive. Forel discovered that if you draw a line through an ant trail with a substance that masks their chemical markers, the ants become confused, lost. their invisible highway erased, but within minutes they reestablished the trail, laying down new scent markers, rebuilding their communication network. He also found that different ant species use different chemical languages, that an ant from one species cannot understand the trails of another, even closely related species. Each colony, each species, operates on its own private frequency, its own secret code. Carl von Frisch, born in 1886, would take the study of insect communication even further. He spent decades studying honeybees, those master navigators and architects. He discovered that bees can see ultraviolet light, wavelengths invisible to human eyes, that flowers appear completely different to a bee, marked with patterns that guide them to nectar like runway lights guiding a plane. He proved that bees use the sun as a compass, that they can navigate even on cloudy days by detecting polarized light in the sky, and most famously, he decoded the waggle dance, that figure-8 movement a foraging bee performs when she returns to the hive. The dance is a map, a set of precise instructions. The angle of the dance relative to the vertical comb indicates the direction to fly relative to the sun. The duration of the waggle portion indicates the distance. A dance lasting 1 second means the food source is about half a mile away. Longer dances indicate greater distances. This was language, symbolic communication, abstract representation in an organism with a brain smaller than a pinhead. Von Frisch's work, published in the 1940s and 50s, shattered assumptions about insect intelligence. He would eventually win the Nobel Prize, one of the few times that prize was awarded for work on invertebrates. Meanwhile, in the early 20th century, entomologists were beginning to understand the true scope of insect diversity. Every expedition to tropical regions returned with thousands of previously unknown species. Estimates suggested that insects might represent more than half of all living species on Earth, perhaps as much as 80%. The sheer number was incomprehensible. Millions of species, each with its own anatomy, its own life cycle, its own ecological role. William Morton Wheeler, an American myrmecologist, argued that an ant colony should be considered a superorganism. The individual ants were like cells in a body, specialized, interdependent, unable to survive alone for long. The colony had a metabolism, consuming resources, producing waste. It had a life cycle. Birth when a new queen established a nest, growth as the colony expanded, reproduction when new queens and males flew out to mate, possibly death if the queen died and was not replaced. This idea that a collective of simple organisms could create something more complex than the sum of its parts influenced fields beyond entomology. It offered insights into organization, cooperation, distributed intelligence. The colony made decisions, chose nest sites, allocated labor, defended territory, but no single ant understood the whole picture. No ant was in charge. The intelligence was emergent, arising from thousands of simple interactions, following simple rules. During the World Wars, entomology took on new urgency. Insects destroyed crops, spread typhus in trenches, threatened armies with disease. The DDT, first synthesized in 1874 but not recognized as an insecticide until 1939, seemed like a miracle. It killed mosquitoes, lice, flies, agricultural pests with extraordinary efficiency and apparent safety. Paul Hermann Müller won the Nobel Prize in 1948 for discovering its insecticidal properties. For a brief moment, it seemed humanity might win the ancient war against insects, eliminate malaria, end crop failures. DDT was sprayed everywhere, in homes, on fields, in massive public health campaigns. And it worked. Malaria was eliminated from entire regions. Crop yields increased. The future looked bright. But Rachel Carson, a marine biologist and writer, was watching the birds. Populations were crashing. Eggs were failing to hatch. Shells too thin to protect the embryos inside. Carson spent years researching, interviewing scientists, examining data, and in 1962 she published Silent Spring. The book documented how DDT accumulated in the food chain, how it persisted in the environment, how it poisoned not just insects, but the birds and fish and mammals that fed on them. The controversy was immediate and fierce. Chemical companies attacked Carson's credentials, her methods, her motives, but the evidence was overwhelming. DDT was indeed persistent, toxic, dangerous. The book launched the modern environmental movement, changed how we thought about pesticides, about unintended consequences, about the interconnectedness of ecosystems. And it reminded entomologists that insects do not exist in isolation, that killing them indiscriminately has ripple effects we are only beginning to understand. Through all of this, through wars and technological revolutions, through changing theories and methodologies, the insects themselves remained constant, adapting, evolving, surviving. In laboratories, they were teaching us about genetics, about disease, about social organization, about flight mechanics and chemical communication, about the fundamental processes of life itself. And in the meadow, our imagined meadow where this journey began, the evening deepens. The moths are flying now, drawn to flowers that bloom white in the darkness, flowers that release their perfume only at night, and the crickets continue their chorus, that sound unchanged since before humans existed, a reminder that we are newcomers to a very old world. The latter half of the 20th century brought technologies that previous generations of entomologists could barely have imagined. Electron microscopes revealed structures at the molecular level, showing how insect wings were constructed scale by scale, how compound eyes were arranged lens by lens. The scanning electron microscope produced images of alien beauty, a mosquito's mouth parts looking like surgical instruments, a butterfly's antenna covered in sensory hairs, like a forest of tiny trees. DNA sequencing, when it finally became possible, revolutionized how we understood insect relationships. Species that looked similar were sometimes only distantly related, while insects that appeared completely different shared recent common ancestors. The evolutionary tree was being redrawn, branch by branch, based not on appearance, but on the genetic code itself, that ancient text written in chains of molecules, telling the story of how life diverged and diversified over hundreds of millions of years. E.O. Wilson, born in 1929, became perhaps the most influential entomologist of the modern era. He began by studying ants, those endlessly fascinating social insects. He and his colleague Bert Hölldobler spent decades documenting ant behavior, communication, ecology. Their book, The Ants, published in 1990, was 800 pages of dense scientific detail, and it won the Pulitzer Prize. Ants became a lens through which to understand evolution, social behavior, chemical communication, ecological dynamics, Wilson identified the concept of island biogeography, working with Robert MacArthur, showing how the size of an island and its distance from the mainland determined how many species could live there. This wasn't just about actual islands. Any isolated habitat, a mountaintop, a forest fragment, a lake, followed the same mathematical relationships. The implications for conservation were profound. As humans fragmented habitats, turning continuous forests into isolated patches, we were creating islands, and those islands could only support so many species. But Wilson's influence extended beyond ants, beyond insects entirely. He popularized the term biodiversity, argued passionately for the importance of preserving species, for understanding ecosystems before we destroyed them. He estimated that we were losing species faster than we could identify them, that tropical forests might contain millions of insect species still unknown to science, and that many would go extinct before they were ever discovered. This realization that we were living through a mass extinction event changed the tone of entomology. The science became more urgent, less about pure curiosity and more about conservation, about understanding what we were losing, about trying to preserve what remained. Entomologists fanned out across the tropics, racing to document species before their habitats disappeared, collecting specimens, recording behaviors, trying to capture snapshots of ecosystems that might not exist in another generation. At the same time, insects were revealing secrets about their own incredible abilities. Scientists discovered that monarch butterflies, those delicate orange and black wanderers, migrated thousands of miles from Canada to specific forests in Mexico, using the sun as a compass, correcting for its movement across the sky throughout the day, using an internal clock to adjust their angle of flight. No individual butterfly made the round trip. The migration took 4 or 5 generations, each one born knowing the route without ever having flown it. The instructions were genetic, inherited, precise enough to guide them to the same trees year after year. Desert locusts, those ancient plagues, were found to transform based on population density. A locust living alone is green, solitary, calm. But when conditions allow populations to explode, when they encounter other locusts frequently, their bodies change, they turn yellow and black, become aggressive, gregarious, form massive swarms that can contain billions of individuals, covering hundreds of square miles, consuming every plant in their path. This was phenotypic plasticity, the same genome producing radically different bodies and behaviors depending on environmental triggers. The molecular biology of metamorphosis was finally understood. Hormones, controlled by day length and temperature, triggered the transformation from larva to pupa to adult. Inside the chrysalis or cocoon, the caterpillar's body essentially dissolved, breaking down into a cellular soup from which the adult structures were assembled, not from scratch but from clusters of cells called imaginal discs that had been present since the larva hatched, waiting dormant for their moment. This wasn't magic, but it was close. A complete reorganization of a living being, death and a resurrection coded into the genome. Fireflies, those living lanterns of summer nights, were found to produce light through a chemical reaction so efficient that it generated almost no heat. Bioluminescence, achieved through the enzyme luciferase combining with luciferin in the presence of oxygen, creating a glow that was nearly 100% efficient. while our best light bulbs wasted most of their energy as heat, and the flashing patterns, those codes of light in the darkness, were species-specific. Each firefly species had its own rhythm, its own signal. Males flashed while flying, females answered from the grass, a conversation in light leading to mating. But some fireflies were deceptive. Females of the genus Photurus learned to mimic the flash patterns of other species. When males of those species approached, expecting a mate, the Photurus female would eat them. This was aggressive mimicry, sexual deception, predation through false advertising. And it worked because the language of light was so precise, so specific, that a correctly timed flash was trusted implicitly. Insects were also teaching us about disease in new ways, not just as carriers, but as subjects for understanding immunity, development, genetics. The fruit fly, Drosophila melanogaster, became one of the most important organisms in biological research. Its genome was sequenced in 2000, revealing 13,000 genes, many of which had direct counterparts in humans. Studying how genes controlled development in flies gave insights into human development, into birth defects, into cancer, into aging. Thomas Hunt. Morgan's work with fruit flies in the early 1900s established that genes were located on chromosomes, that they were inherited according to mathematical laws. His fly room at Columbia University, crowded with milk bottles full of flies, produced discoveries that earned him the Nobel Prize in 1933. And fruit flies continued to be invaluable, easy to raise, quick to reproduce, with giant chromosomes in their salivary glands that could be seen under basic microscopes. They were the perfect model organism. Meanwhile, in the wild, entomologists were discovering that insect populations were declining, not just rare species, but common ones. Insects that had been abundant were becoming scarce. Long-term studies in Europe showed drops of 75% or more in flying insect biomass over just a few decades. The causes were multiple: habitat loss, pesticides, climate change, light pollution disrupting nocturnal species, death by 1,000 cuts. The implications were staggering. Insects pollinate roughly 85% of flowering plants. They break down dead matter, recycle nutrients, form the base of food chains, feed birds and fish and small mammals. If insect populations collapsed, ecosystems would collapse. We were discovering their importance precisely as we were losing them. The irony was bitter. Some entomologists turned to citizen science, engaging the public in monitoring butterfly populations, counting bees, documenting fireflies. The data was valuable, but more than that, it reconnected people with insects, encouraged them to look closely, to notice what was in their own backyards. Children who might never have thought about insects were suddenly keeping records, learning species names, understanding life cycles, and technology offered new tools for conservation. Genetic analysis could identify illegal trade in protected butterflies, track disease outbreaks in bee populations, determine whether populations were isolated or connected. Drones could survey remote habitats. Automated cameras could monitor rare species. The same technologies that had contributed to environmental damage were being turned toward protection. In laboratories, researchers were exploring how to use insects to solve human problems. Flies were being studied for their extraordinary navigational abilities. their compound eyes and rapid processing inspiring new designs for motion detection sensors, beetle shells, with their structural colors that needed no pigment, inspired new materials, new paints that would never fade, the water-repellent surface of the Namib desert beetle, which collected fog from the air in one of the driest places on Earth, led to new water harvesting technologies. Ants, with their ability to find efficient paths through complex terrain, inspired algorithms for routing internet traffic, for optimizing delivery routes. Their collective decision-making, choosing nest sites through a distributed voting process, offered models for artificial intelligence for understanding how simple rules could produce complex emergent behaviors. And still, despite centuries of study, despite electron microscopes and gene sequencing and supercomputers running models, insects retained their mysteries. New species were being discovered constantly, not just in remote jungles, but in cities, in well-studied forests, in collections where they had been sitting, misidentified. For decades, each new species raised new questions. How did it make its living? What did it eat? What ate it? How did it fit into its ecosystem? The more we learned, the more we realized how much we didn't know, how many species existed, how many were going extinct before we found them, how ecosystems would respond to their loss, whether populations could recover, whether we could change course before the damage became irreversible. In the meadow, the night deepens further, the moths continue their flight, the crickets their song, and beneath the soil, in chambers and tunnels we cannot see, larvae continue their slow transformation, feeding in darkness, preparing for wings they do not yet possess, for lives they cannot yet imagine. The ancient patterns continue, indifferent to our attention, indifferent to our understanding or lack of it. The insects simply are, doing what they have done for 400 million years, surviving, adapting, filling every available niche with an exuberance and diversity that still, after all this time, takes the breath away. We find ourselves now, not at the end of the story, but at a place of deeper contemplation, standing in the meadow as true darkness settles, looking not just at what insects are or what they have been, but at what they mean, what they reveal about the nature of life itself. Consider the scales of time at work here. A mayfly lives for a single day as an adult, emerging at dawn, mating, laying eggs, dying before sunset, its entire purpose compressed into hours, and yet the nymph stage beneath the water might have lasted a year, 2 years, even 3. Most of its life spent in preparation for a single day of flight. Is this tragedy or efficiency or simply a different way of organizing existence? A cicada of certain species spends 17 years underground, feeding on tree roots, growing slowly in darkness, waiting for a signal we still don't fully understand. And then, in a single coordinated emergence, millions of them surface at once, climb trees, split their skins, unfurl wings, fill the air with sound, mate, and die within weeks. 17 years of waiting for one brief moment of noise and reproduction. The mathematics of this are perfect. 17 is a prime number, making it harder for predators to synchronize their own life cycles to exploit the emergence. These are not conscious decisions. These insects do not choose their strategies, and yet the strategies have worked for millions of years, refined through countless generations, through the brutal editing process of natural selection, where every inefficiency is eventually eliminated, what remains is elegant, even when it seems strange to our eyes, even when it defies what we might consider a good life. The hive mind of bees and ants troubles our assumptions about individuality. We celebrate autonomy, independence, the lone hero making choices, but these insects suggest a different model. The colony as the unit of selection, the individual as expendable. When a honeybee stings a mammal, her barbed stinger tears from her body, and she dies. This makes no sense from an individual perspective, suicide as defense, but for the colony, it makes perfect sense. One bee dies, the intruder is driven away, the hive survives. The bee's sisters, who share 75% of her genes, live to reproduce. From the gene's perspective, the sacrifice is logical, even generous. This challenges our comfortable notions of self, suggests that perhaps individuality is not the only path, not necessarily the best path, that cooperation at the cost of autonomy can produce something remarkable, cities of insects underground, chambers and tunnels, nurseries and food storage, agriculture. Some ants farm fungus, some aphids are herded like cattle, all this from creatures with brains smaller than a grain of sand. No individual ant understands the whole, but the whole works, thrives, persists. And what a beauty, that word we use so carefully, so subjectively, the butterfly emerging from its chrysalis, wings crumpled and wet, pumping fluid through the veins, watching those wings expand and harden, the pattern revealing itself, orange and black, or blue iridescent, or brown with eye spots that startle predators. This beauty serves a function. Everything serves a function. The colors warn of toxicity, or attract mates, or provide camouflage. There is no art for art's sake in nature. And yet, we cannot help but see it as beautiful. The scarab beetle's metallic sheen, the dragonfly's transparent wings, the firefly's gentle glow. These things move us, speak to something in us that recognizes pattern, symmetry, elegance. Perhaps beauty is not frivolous after all. Perhaps it is the recognition of efficiency, of solutions that work so well they take on an aesthetic dimension, the beauty of mathematics, of physics, embodied in chitin and scales and bioluminescence. The ecological web that insects weave is so complex that we are still mapping it, discovering it. A single oak tree might support hundreds of species of insects, each with its own niche, some eating leaves, and some eating bark, some eating other insects. And those insects feed birds, feed bats, feed spiders and lizards and frogs. The tree is not just a tree, it is a universe. And when we lose a tree or a forest, we lose not just wood, but entire worlds, countless relationships, dependencies we never knew existed. There is something humbling in this, in realizing that the small things, the overlooked things, are not peripheral but central, that we mammals, large and warm-blooded and proud, are the minority, the exception, that the real success story of terrestrial life is written in 6 legs and compound eyes, in metamorphosis and chemical communication, in societies older than our species. Some estimates suggest there are 10 quintillion insects alive at any given moment. That is 10 billion billion, a number so large it becomes abstract, meaningless, and yet each one is an individual with its own brief moment of existence, its own struggle to eat, to avoid being eaten, to reproduce. each one a tiny expression of the same force that drives all life, the blind imperative to continue, to persist, to pass genes forward into an unknowable future. And they do this without fanfare, without expectation of recognition or reward. A dung beetle rolling its sphere across the sand does not know it is recycling nutrients, returning nitrogen to the soil. It knows only the imperative coded in its neurons: roll the ball, bury the ball, lay the egg. The function emerges from the action, not from understanding. This, too, is instructive. We who overthink, who plan and worry and second-guess, might learn something from the insect's certainty, its commitment to the task at hand, however strange that task might appear to an outside observer. There is a kind of freedom in having no choice, in being what you are without question or doubt. The meadow around us has gone fully dark now. The last light faded from the western sky some time ago, replaced by stars, by the cool silver of a moon not quite full, and the insects of the night are in their element, owning the darkness, navigating by scents and sounds and the faint movements of air. Somewhere a moth finds a flower, uncoils its long proboscis, drinks nectar that tastes of specific sugars, specific compounds. It will fly to another flower, carrying pollen on its fuzzy body, facilitating reproduction for a plant that bloomed specifically for this moment. For these nocturnal visitors, the relationship is ancient, specific, essential. Neither moth nor flower chose this. They inherited it, received it from ancestors stretching back through deep time. The cricket's song continues, will continue through the night, males calling, hoping for a response. The frequency of their chirping changes with temperature, slowing as the air cools. You could calculate the temperature by counting chirps. By knowing the mathematical relationship between heat and muscle contraction, between energy and sound, everything connects, everything relates, through principles of physics and chemistry and biology, all operating at once, in the soil beneath our feet, in the leaf litter and the roots. Larvae continue their transformations, beetles forming in pupal chambers, their adult bodies taking shape, folding and compressing, waiting for the signal that says, it is time to dig upward, time to emerge, time to begin the brief adult phase with its imperatives of feeding and mating and dispersing. And in the tree above, a colony of ants settles for the night, activity slowing but never stopping entirely. Some workers still patrol, still tend the brood, still maintain the nest. The colony breathes, if we can use that word, cycles of activity and rest, but never complete stillness, always some subset awake, alert, ready. This has been happening here, in this meadow and places like it, for longer than we can properly comprehend. Long before humans, long before mammals, long before the first flower bloomed. Insects were here, different forms perhaps, different species certainly, but recognizable. Six-legged and segmented, compound-eyed and antennaed, the basic body plan proven successful, maintained across hundreds of millions of years of change, and it will continue after us. This seems certain. Insects survived the extinction that took the dinosaurs, survived ice ages and volcanic winters and asteroid impacts. They are resilient in ways large animals are not, small enough to hide, numerous enough to endure catastrophic losses and recover, diverse enough that some species will survive almost any calamity. They will be here in some form long after we are gone, still pollinating, still decomposing, still filling the air with their humming and the night with their song. There is comfort in this continuity, in knowing that some patterns are too old, too tested, too fundamental to break easily. The metamorphosis will continue. The emergence will continue. The ancient dance of predator and prey, of pollinator and flower, of parent and offspring will continue. We are privileged to witness it, to study it, to begin to understand the complexity and beauty of these small lives. And perhaps, if we are wise, we will learn to live alongside them more gently, to preserve the habitats they need, to make room in our world for their world, to recognize that their flourishing and ours are not separate things, but intertwined, dependent, necessary to each other in ways we are still discovering. The night deepens. The meadow settles into its rhythms, and somewhere in the grass or in the trees or beneath the soil, a life is beginning, a transformation occurring, a tiny drama playing out with no audience but the stars. And this is enough. This has always been enough. Life continuing, adapting, persisting, in forms small and strange and utterly essential. The insects carry on, as they have always carried on, and the world turns, and the seasons cycle, and the ancient patterns hold, and we can simply stand here in the darkness, listening to the crickets, watching for the occasional firefly, breathing the cool night air, grateful to be part of this vast living tapestry, this planetary experiment in diversity and resilience, this beautiful, endlessly complex, quietly miraculous world